All right, so this will be the last lab manual or fourth lab unit for the semester. This unit is focusing on urinary system anatomy, reproductive system anatomy, and the histology of urinary and reproductive systems. We're not going to be doing a rat dissection. It's unfortunate, of course. It's a nice way to sort of end the semester, seeing lots of different organs in the rat, but I left those slides there in case there's a nice video there and some other information about it if you're curious and kind of want to check it out, but I won't be testing you on the rat dissection. So I just want to run through this as typical. Like by now you guys pretty much know how these work, but I just want to go ahead and highlight some of the things, and especially the histology. I want to go over some specifics there and make sure everyone's clear on how that works. All right, so first slide here is just some background on the urinary system, and then moving on to the kidneys and identifying the different structures and location and surface features of the kidneys. Of course, as typical, you just want to be able to identify the things in the boxes that are listed there, and those are the items that I will be testing you on. So this image here is a nice sort of location in terms of where the kidneys are found in retroperitoneal, which means sort of the back wall of that peritoneal cavity. Of course, the adrenal glands sit on top of them. You can see a lot of the blood vessels that feed into the kidneys. That's a significant role in terms of how the kidneys work, is the blood flow. So we'll go over those as well. All right, so in terms of moving further with the kidneys, you want to be able to identify the internal anatomy and all the structures that are listed here. And this picture is an excellent representation of a cross-section, or not a cross-section, but a sectional frontal section through the left kidney. And what you can see are the two primary locations of the kidney in the inside. You have what's called the renal cortex and the renal medulla. The renal cortex or the cortices are where actual initial production of urine occurs, and what are called nephrons, and we'll go over those in a minute. But things like renal pyramids and lobes, and these all feed into what are called major and minor calyces. So you want to make sure you're aware of those. And essentially, all these different sections of these renal pyramids ultimately feed into the renal pelvis, and those then drain into the ureter, which of course then leaves the kidneys and heads down to the urinary bladder, which is a temporary storage location. So I want to hit all those. That's a good picture for that. Next is want to move on to more of the microscopic anatomy of the kidneys, and this is what's called a nephron. A nephron contains a renal corpuscle and a renal tubule, but also within that what's called the glomerulus. And the glomerulus is the structure, or the capsule, Bowman's capsule, where urine formation initially occurs. So we want to be able to identify all those different structures. Here's a nice illustration of blood flow entering and leaving the nephron, but also the tubular system, which is essentially where all the further reabsorption and filtering of the urine occurs. So that's a nice one to sort of view the overview of the whole nephron. This moves specifically onto the corpuscle or the glomerulus. And this is a great image because it shows sort of where blood enters into the glomerulus, which is what's called an afferent arteriole. And it moves through the structure, which, as you learn in lecture, is more of a fenestrated capillary lining that allows for fluids to be forced essentially by hydrostatic pressure into this capsular space, and that's the initial formation of urine that we call filtrate occurs in that location. But then, of course, it goes through the proximal convoluted tubules, the loop of Henle, the distal convoluted tubule, and ultimately goes through the sort of further processing and fine-tuning of what will ultimately be the final product that will leave the body as urine. So that's a really nice slide. You want to make sure you have that. Another really important part of the kidneys is blood flow, as I mentioned. And so kind of like you did blood flow with the vessels chapter, as well as with the heart, you're identifying these different vessels. But essentially, as they branch, they get smaller and smaller, but they also then take on a different name. So you want to sort of have that branching and be familiar with that of all these renal blood vessels. So this is a great shot of a sectional view, again, focusing more on the blood vessels. So it starts at the renal artery and then branches into the segmental and then to the interlobar and the cortical radiate, so forth and so on. So you want to be able to identify those. So that's a good slide for that. And then we can zoom in just on a renal lobe, one single renal lobe, and you can see branching even further as it leads up to the glomerulus. So this is another really good shot of those structures. This shows the pathway of blood flow through, starting at the renal artery, moving through the nephron, and then exiting back out the renal vein. So these are the vessels that you'd be learning, and this is just sort of another way, I think, to kind of visualize the progression or the branching that occurs as it flows through the renal circulation. So that might be useful for you to identify, or to understand that flow is going to help you identify those structures in the image. All right, so that's the kidney. Then you move on to the ureters. Ureters you can kind of think of as, if you want a reasonable correlate, would be thinking about the esophagus going from your oral cavity to your stomach. It's kind of what the ureters do. They're coming from the kidneys where the initial production is occurring, and it's a muscular tube connecting the kidneys to the urinary bladder. And the urinary bladder, as you know, is really a temporary storage location for urine until as it fills. We'll see how that micturition reflex allows for the release of the urine out the urethra. So we'll look at that coming up as well also. So the ureters, you want to know that. Here is a cross-sectional view of a ureter. Probably don't need to know that one, but at least it gives you a nice idea of what the histology section is looking like. And it's fairly similar to the bladder in that it has transitional epithelium with smooth muscle to sort of push those fluids down to the bladder. So that can be useful just to get an idea of what the ureters are made of. All right, then that leads down to the urinary bladder, and so there are a lot of different structures within the urinary bladder that you want to be able to identify as listed here in this box. And this is a great picture, of course, that I will use to have you identify a lot of the structures. This happens to be a urinary bladder of the male, and you can tell because there's a prostate gland, which is only in the male. We'll talk about that in a minute with reproductive system. But you can see a lot of the different structures from the different ligaments and the ureter, the trigone, which is sort of like a funnel for the urine at the bottom or at the base of the bladder. So you can see all those images there. That then leads on to the urethra, and the urethra is different for male and females. Essentially with the male, it's much longer and has 3 different divisions, mostly based on what it's passing through. So you have what's called a prostatic urethra, because part of the urethra actually passes through the prostate gland, membranous urethra, and then a spongy urethra, and then ultimately the external urethral orifice. So you want to have all those, of course, and there's a nice picture showing all those different structures. And some of this stuff will overlap with the male reproductive pathway also. So we'll get that slide in a minute. Then you have the female urethra. Primary difference, again, as I was mentioning before, it's much shorter, only 3 to 5 centimeters from the bladder to the vestibule. And then it's not passing through these larger glands that we saw, like with the prostate gland in the male pathway, obviously. So that different structures you want to know for the female urethra. And there's a nice image for that. Again, same with the female reproductive pathway. This image will cross over with some of those images that we'll see in a minute. All right, so that's it for urinary. So you have kidneys, ureters, your urinary bladder, urethra, all the different blood vessels associated with the kidneys. I want to make sure you have all that. Then we move on to the male and female reproductive systems. Male reproductive system, we're going to start with the testes. And these images are actually probably better than some of the models we have, so we probably would have used some of these images in lab anyway. But things like the epididymis and the ductus deferens, the seminiferous tubules, some of those things are just easier seen on the actual illustration that we have here. So this is a nice picture showing a lot of those structures. Again, this is similar to what we saw in the urinary pathway. And so you can sort of zoom it in. You can see the seminal gland, the prostate gland, the vulvourethral glands on this, which are responsible for producing semen. And then here's another shot where you can see those same three glands as well as the urinary bladder with the male. And you can also see the prostatic urethra in this image. So that's a good shot for those structures and those glands. The external genitalia you can see in this picture, that works fine for identifying those structures. You can see this. And this image structure of the testes is really useful because it's a cross section through the testes. And what you can see nicely here are the seminiferous tubules, which are these coils of tubules that you see, and that's where sperm production occurs. So we'll be looking at these seminiferous tubules when we do the histology section, but that's sort of a visual of where those are located within the testes. So those are an important structure, seminiferous tubules that you can see, and those lead into ultimately the vas deferens via the epididymis. So we want to make sure you have those understood, and that'd be a good image, or at least a sectional image of those seminiferous tubules for that. Female reproductive system, starting with the ovaries. We'll also be looking at the ovaries in the histological section, but here is just sort of an illustrative picture with its relationship, the ovary relationship to the uterine tubes as well as the uterus, which you can see in that image. That, of course, then, as I said, leads into the fallopian tubes or uterine tubes, where you see different regions of the uterine tubes. As I said, you have the fimbriae, which are attached essentially to the ovaries, and that leads into the infundibulum, then the ampulla and isthmus. And as we'll see in lecture, these regions are important because ultimately, in the proximate location between the isthmus and the ampulla is where we're going to see fertilization occur if it's going to happen. So that's important to be able to recognize those structures. On to uterus. Want to be able to understand the different regions, of course, of the uterus, but also the different layers from a histological section, parametrium, myometrium, and endometrium. And we have slides of that. This is a really nice cross section where you can really see all of it. You can see the ovaries, you can see the fallopian tubes, and you can also see the uterine cavity and the thick uterus muscular wall. Identifying those structures would be important. The next location is the vagina, where we have the fornix and the stratified squamous epithelium. And we also have some external genitalia, which you can see in these next two slides. So this would be a good slide for the vaginal canal and other associated structures. And then finally, on to the mammary glands for the female reproductive system. Primarily what we're going to be looking at here is this image where we can see different ductual structures and different lobes, and the fat pad that's part of that as well. So all those structures you can see nicely in this image at this time. All right, so last thing then we want to look at for this next unit is the histological section of all these different tissues, as I was mentioning before at the outset. And so this we haven't really been over too much, so I want to make sure you have a good idea of what we'll be identifying histologically, as well as some of the different sites you're going to be looking for. So first thing you want to look at and identify here are the kidneys. The kidneys, what we're looking at, if you recall from the previous images that we looked at with the kidneys, we saw the renal cortex. And so this tissue sample is taken from the renal cortex. And so what you can see nicely, as you know, that's in the renal cortex are these structures here, which are the renal corpuscles. And the renal corpuscle, of course, contains the glomerulus, which are these guys, and then the capsular space, of course, which is there. And so, as we talked about previously, and as we've talked about in lecture, the blood flow moves through this glomerulus, and essentially, with the fenestrated capillaries of these glomerular structures, forces the fluid into that capsular space, and you can see that nicely on this histological section. Also, the tubular component or compartment of the renal cortex is where, as I said, all sort of the fine processing of the urine occurs, and that's made of simple cuboidal epithelium. And you can see all these simple cuboidal epithelial cells sort of scattered about in this section. So that would be what you need to identify for the kidney. So just like we've done previously, you need to tell me which organs tissue sample comes from, and then any of the different sites or specific things that I mentioned, you want to be able to identify those as well. All right, next on the urinary bladder, what we're looking at is a couple things primarily that I want you to be able to identify. The mucosa, which is, of course, the transitional epithelium, which is up here at the top. You guys know about transitional epithelium from your studies in A&P 1. Transitional epithelium has the ability to expand and recoil, right? The cells sort of change size and shape depending on, with the bladder, how much fluid is in the bladder. So that's what you want to see in terms of mucosa. The detrusor muscle is basically smooth muscle, very large band of smooth muscle that allows, as it stretches, of course, it wants to contract. We talked about that as it relates to the micturition reflex. But that's where that's located. And then rugae is just sort of the undulating surface that we see of the bladder. So it sort of just moves in kind of an up and down pattern, of course, allowing it for it to stretch out or to recoil. So rugae would be another thing that you could see on the urinary bladder. Next one is the testes. So this is a cross-section, histological cross-section through a seminiferous tubule. So this essential circular section you're looking at here is one seminiferous tubule. And if you recall the seminiferous tubules, this is where sperm production, essentially, and maturation, not complete maturation, but at least initial formation and maturation occurs in this location. And so a couple of features you want to have. First of all, the deepest layer of cells that you can see is what's known as the germinal epithelium. And so kind of like we talked about with the skin, for instance, right? These are where we have essentially stem cells that are dividing and producing what are called spermatogonia. And spermatogonia are ultimately going to go through a series of developmental processes. And once they get to the lumen, which is this part here, right, they reach a certain level of maturity, then the sperm will be released into this tubule pathway, and they can move on through the tubes all the way up to the epididymis, and then ultimately to the vas deferens, when it would be ultimately potentially to be released. So those are the types of things you want to be able to identify within the testes as it relates to the seminiferous tubules. Next, we have spermatozoa. These are a histological section of a couple of mature sperm. And what you're looking to identify here are a few things. Number one is what's called the acrosomal head. Okay, and this is sort of that white kind of tip on the head of the sperm. And that's really important. If you recall from lecture, we talked about how during the formation development of sperm, the Golgi apparatus, which is typically responsible for secretions in most cells, sort of reorganizes and reconstitutes at the top of the head of the sperm and forms what's called an acrosome. And ultimately, that acrosome is really important in fertilization because, as you'll see when we look at the ovary and look at a secondary oocyte, the oocyte itself is surrounded by an antrum as well as a corona radiata and other structures, zona pellucida, that sort of protects it essentially. And so when the sperm is trying to fertilize the oocyte, it needs to sort of burrow through those linings, which we'll see on the next slide. And that acrosomal head is important for that to be released in that location. The nucleus, of course, the nuclear content will be inside the sperm. So that's all the genetic material that the sperm is going to bring to the fertilization process. And then you have next what's called a mitochondrial spiral, which is this section here. As you know, again, the mitochondria of these cells as they're developing need to reorganize, and they need a lot of energy. Sperm actually have this flagellan corkscrew tail at the end where they can actually swim, but that requires a lot of energy, utilizing the sugar that's found in semen to propel them forward. So that's the mitochondrial spiral. And then, of course, we have the tail here at the very end there. So those are different features. You want to be able to identify the sperm. And then finally, we have the ovary. What we're looking at, I've zoomed in on one specific section of the ovary, and we're looking at what's called a tertiary follicle. So this whole thing is a tertiary follicle, and this structure here in the center here where you see this nucleus is the secondary oocyte. So the secondary oocyte is sort of suspended within this tertiary follicle, and in this tertiary follicle you have this antrum, which is here, which contains follicular fluid. And then what happens is at ovulation, essentially the secondary oocyte, including the zona pellucida and the corona radiata, are released from the ovary and ovulated into the fallopian tube. And so that's the structures that I was talking about. The corona radiata is sort of this crown of cells here. And then the zona pellucida is kind of this black line that you see encircling the secondary oocyte. So those things are all shed from the tertiary follicle at ovulation. And so those different structures you want to be able to identify as well as it relates to the ovary. All right, so that's it on the histological sections. As I said, the last part, the rat dissection, you're not, responsible for. I'm not going to be testing you on that. But if you want to watch, there's a nice video here showing that. I've also included some, the information about how you would go about doing the rat dissection and the different things that you would be finding when you did your rat dissection. So if you're curious about it and want to kind of read over it, you're welcome to. But essentially, you would be identifying a lot of major organs like the heart and the lungs and the trachea and the kidneys and the stomach and the small intestine, so forth and so on, as well as the urogenital structures of the rat in both the male and female. So it's unfortunate we can't do that this time, but that's sort of the limitation of where we're at currently. All right, and then again, I've just included a couple other pictures as well if you want to peruse those. So that wraps up this fourth lab unit. And that will do it for the semester. Just email me with questions.